Brazil. But today, for the next around 40 minutes, right, you will talk about sorting, nano-sorting, Cytoflex SRT, and like just a normal day in core facility. That's the topic we will talk today. So let me share with people some information about this live. After the presentation, we will have time for questions and answers. But during the presentation, you can add your question in the chat. And please, everybody, feel free to write in Portuguese or in Spanish if you feel more comfortable in write your questions in your language, local language. And I can translate your questions to John. And important information for whom don't have YouTube account, we will see email from Beckman Coulter. You can send your question through this email, and we can read your question after John's presentation. Okay. Okay. Let me present officially John Tigges for you, and I will give the word for him. Just before present John Tigges again, thank Flowmentor again. This channel is Flowmentor YouTube channel, and we have to thank them to open this space for us. So let me present officially John Tigges. He is Principal Associate in Medicine at Harvard Medical School and the Director of the Flow Cytometry SRL at Beth Israel, the Cones Medical Center. His current interests include vaccine research and nanoflow cytometry. Thank you again, John, and you have around an hour to speak, and we'll be here to hear you.
Thank you very much, Raquel. Thank you, Flowmentor, as you said, and everyone who's joining us today. As I was telling Raquel, I will try to be cognizant of my cadence, as I know that I may be one of the few naturally English-speaking individuals on this today. So again, if there is something that may not be clear, please write it down in the question, and I'll try to go back and clarify as much as possible. So the first things I need to do here is make sure that I do this little slide so that everybody can see it and know that what I am saying today is what is in my core, what I do on a daily basis, and how I use the instrumentation that is within my core and I have availability to use. Also, be aware that I am a research core facility. While we may be involved in some preclinical or clinical type data, this is not necessarily what we'll be talking about today. And so the instrumentation that we will be mentioning and the projects are all in research, and the machines are research use only. Okay, now that we got the legalities out of the way, let's talk about a little bit about this, right? So when this came out, I believe it was like last March, or maybe the March previous. Anyways, I had one of these a little bit previous to that. So it's coming up on almost 3 years. Been involved in the Cytoflex SRT project. And basically it is the Cytoflex that sorts. They're not kind of lying there. There is also this portion here where we have the picture, but just to give a better perspective, this is actually the same size as the LX. So the LX and the Cytoflex SRT are pretty much the same size. So benchtop sorter, yes, it is. Some of the features that we have on this are, that make it a great platform for us in our hands is that we had the Cytoflex S, we have the LX, along with other instrumentation, but they become very complementary. That you can start doing your discovery panels on the Cytoflex analyzers and easily move this over to the sorting platform. And I put the S here because that's what it was built off of, right? The SRT was built off of the S platform. They have put in many wizards. So those of you that are used to BD instruments or SciTech instruments where there's wizard bases that everything starts up and goes through processes and makes it very kind of intuitive. The same thing was used here. Now, this is one of those things that's going to be very kind of great, but also controversial, right? So gentle sorting, 100 micron nozzle tip. Not the gentle part, not the 15 PSI, it's that 100 micron. I know there's such a call for the ability to have a 70 micron, but this was built in the thought process of how can we make the cells happy and keep them happy. And so by reducing that pressure and using a larger nozzle size, the applications that you can do just become extremely vast and broad. So for us, what we use it for and what we're doing, it is perfect. We have other things using 70 micron nozzle tips for some of our nuclei work, but this is great for most applications. And saying that, it's great for rare event visualization, index sorting. The volume is not terrible considering it's a 100 micron nozzle tip. Because of the lower pressure, you actually generate a little bit smaller droplet than you would normally expect, hence less volume. And the thing we'll kind of touch base on, and I know a lot of people are super interested, is that violet side scatter that is well known on the Cytoflex platform, and how does that translate into the SRT or the sorting platform? Just quickly, a list of applications. Obviously, we all have many things that we like to do. You may have a specialty. If you're part of a core facility in any sense, whether flow cytometry or omics or doing small animal imaging, you have a vast array of things that you need to kind of have knowledge of and be able to work with. That's just a basic idea of things that we're going to touch on today. And that is not exhaustive by any means. There are tons of other things that we do. Some of them, the simplistic GFP sortings to, more of the complex, type assays will show a little bit of that. So again, we're saying the complementary platforms, you don't need to take my word for it. I put it on a slide, so you didn't have to. So you can see here, the optical benches are exact. So everything in the laser lines and the way that the beams are focused to the flow cell and the interrogation point are all the same, along with your actual band passes here and how you can change those filters. They're interchangeable, and we'll go into actually how that looks in a caricature or in artist rendering, probably better. So here you see the optical filters. You've got the APDs, the fiber optic cable that's bringing in the laser light, the mirror which will bounce the laser light across, as you can see here. And as the Cytoflex platform is known for, is higher intensity signals due to that avalanche photodiode and the lack of real laser light loss, as you can see in there, where it bounces back and forth and it finds where it is actually accepted through that filter. And it just kind of bounces back and forth on the mirror so that you get as much laser light as possible into the avalanche photodiode, which tends to be slightly more sensitive. And here again, just so it's not me telling you these things, this is adapted from a Hamamatsu diagram that they have showing how an APD, EMT, and how the two of them relate in senses of what wavelengths they see best. As you can see, as you get to the lower wavelengths into the UV, they tend to even out. Maybe the PMT would show a little bit better. But you get a nice kind of plateau on the APD between what would be the visible and a little bit of the infrared laser lines. And so this is a kind of advantage above the traditional flow. This is what the Cytoflex platform has been built on in its backbone. So you can see the quantum efficiency rises, and this is why we can get better signaling. Again, some of you may already know this, dealing with the Cytoflex or even in other instruments that may use APD based, or now very equivalent to a silicon PMT. So, optics have really been increased and getting smaller over the years. I say complementary. Right within the software, it was a very smart idea of someone on the team to look and say, okay, in the software, let's just put a thing that says convert site expert experiment. This means you take your Cytoflex S, you do your discovery panels where you find a population of interest. You build this panel, you look, you dig deep, and maybe you've done some other analysis with orthogonal platforms, and you find, no, this is really, really what we want. You can move that experiment over to the SRT without having to rebuild it or put the information in. It's one-to-one, converts right over, and you can get very good data. Here, we're just trying to show a wonderful workflow and how that would assist you to make your life easier. Because everything that you do to make easier and faster is better, because you're thinking to yourself, why? Well, if you're first there to apply for a grant, better chances of getting the grant. If you're first there to publish a paper, well, then your PI and yourself are very happy and proud. So everything that can get you to your destination a little bit quicker is really advantageous. All right, some of the other features, what we're going to show. So if you haven't seen an SRT, if it's not in front of you, you haven't gotten to play with it, you haven't explored the website, just going to give you what we kind of like about it. Getting it ready and going is very simple. Everything's automated. Obviously, setting up your sort and sorting is not automated per se. You need user input. I think if there was no user input, we'd be a lot more upset. I like to be able to touch things, so even some of this automation kind of gets me where I'm not the happiest, but let's just go through it. Startup's pretty easy. A wizard again, put the nozzle tip in, hit this button. It goes through all of its different pressurize, diagnose, debubble, stabilize, and then asks you if you want to start QC. And then basically the QC, you take the same type beads that you have for the Cytoflex, you load them in, it runs through, sets all the delays, sets all the parameters, and gives you a report. These reports are all saved. You can do the Living Jennings plots on those. Anything that you need to transfer that information to another individual or for yourself for record keeping. The calibration, the same thing. It's using similar to the MoFlow type IntelliSort. So it's going through, it's looking at the top and the bottom of the neck, looking for the break off, and then it's determining that stream calibration. It will then set a drop delay along with the phase and the voltage for the side stream. So all this is automated, and all of it comes to you very easily. The other thing, I don't know how easy it is to see, but every time you get a nozzle tip, it comes along of the recommended frequency and amplitude. So this helps in being able to, you have a manual drop calibration tag here. If you hit that, you can actually type those numbers in and kind of give the system a place to start and find. And you'll find that this is pretty accurate. So it was very surprising that this was part of the whole platform, in that this is not something that I've ever seen before where nozzle tips are tested and actual frequencies and amplitudes are kind of dialed in. So that was a very surprising and also welcome addition to this. And I wasn't aware of it first. I just kind of put in my nozzle tip and went. And when I realized why they wrote that on the on the holder, it was very enlightening. And the final step is when you're done and you shut down. It's a simple hit the button. It will go through a process. And this is pretty automated. You can go and grab yourself a cup of coffee, or even tell somebody else, hey, can you just hit the yeah, I'm finished button, close. So that's the entire automation, maybe 30 minutes that it would take. So it's pretty fast, pretty accurate. And if you have a remote connection, you can do this from the comfort of your own living room. So this is the only one that gives me a little trouble getting over because of all that automation. There we go. The other thing is just to touch base, and I was showing this slideshow to my colleague Garrett, and was wondering if I should take this actual slide out. And he said, no, people will be very interested in this top section there. where you can pick what tube size you're actually putting into your collection, whether it's a 5 ml, a 15 ml, whatever that collection tube is. And even if it doesn't give it as an option, you can, select an option and then tell it, what is the initial volume? So that's what these numbers of total volume and percentage down at the bottom is you've told it how much you have in there. It knows when it sorts what the droplet size is, and it's going to allow for a certain amount to be put in. That you've asked for too many events according to the fluid that you have in there, it will actually alert you when you type that number in, what you would like for a sort count, and say, it may not fit in the tube. So this is kind of another wonderful thing where you can kind of calculate out how much volume it would take and say, okay, I need to come back at a certain amount of time and actually change out the tube. Again, you can do this from, I guess your break room or whatever kind of makes you happy. So there's the tip. It's kind of like the new car keys, where it looks just kind of like those little receptor ones. We no longer have to put them in, but this one does go in. I will tell you, it's almost impossible to lose because of its actual size, and I believe that's pretty much to scale. And so even though the actual nozzle portion is very small, it is a nice thing, because I've lost more than my share of those small ceramic nozzle tips. And at the cost they are, that's not a good thing to do. All right, so this is where I'm also going to go back to the why the 100, why not the 70? What would happen? Well, maybe the 70's in the cards for the future, but this is the thought process. And I know the thought process because I was there during the, voice of the customer, key opinion leader type thing. And the whole thing was looking at omics, and especially genomics and 10x particularly. So we moved right up the line where 10x was the goal, as 10x genomics owns 80 to 85% of the market share of everything you do in your, genomic sequencing, single cell seq and whatnot. And right from their webpage, it tells you right out, we recommend. It tells you use a larger nozzle, okay, 100 micron nozzle. Use lower pressure. Well, all right, the SRT has lower pressure. So you can preserve the cell health, you can make sure that fragile cells aren't broken up, and you can get a better outcome on your omics. Well, they also tell you right here that, well, sometimes you got to increase the flow rate because maybe your cells will die before you can get them to our assay. And I guess that's them covering themselves. But I said, well, that's great. So let me take some CAR T cells. These tend to be very kind of fragile. They will die kind of cells. And I'm going to sort them at different rates. So I took it and I went, well, I'm going to sort them at 1,000 events per second. I'm going to sort them at 4,000 and at 11,000 events per second. And what I was actually looking for, and as you can see here, is, does it change too much? Do I get a high coincidence rate if I go too fast? Do I lose a bunch of cells if I go too fast or too slow? And two things I did find out, and one of them was what I was looking for, which was, hey, where is the actual availability of this? Can you go fast? Can you go slow? And do you get the same thing? And as you can see, we're not losing any kind of fluorescence. The mean fluorescence intensity changes slightly. But the second thing it showed me, which I was not anticipating, was looking at, okay, I found an actual optimized speed for these particular cells where I get a great recovery, a great yield, and my fluorescence intensity is maximized, or my MFI. So this was something I was not looking for, but thought, oh, wow, this is an added thing that I learned from doing this very simplistic experiment. And when we did post-sort viability, all of them were equal. So the couple of times that we've done it, whether we got viability that was 90% of the CAR T cells, or we got viability for that particular lot that was 75%, it stayed consistent within all the sort speeds. Another wonderful feature, index sorting and rare event visualization. This is great for, many of the things that I'll show you here today, and especially rare events, when sometimes, which we'll talk about extracellular vesicles, if you're talking about fluorescence on an extracellular vesicle, talk about your rare event. These are, your basic epitopes that would sit on one of these is, even at best case scenario, you're looking at maybe the 120 of a MESF. So your equivocal soluble fluorochrome is extremely low. And this would be calculated out on a 120 nanometer vesicle that you're basically putting epitopes, kind of right next to each other, cheek to cheek, and optimizing it. So this calculation was run through and again, confirmed by using the Solarcus kit and things that John Nolan had seen. So here we're just looking, this is just a simple 8-color immune panel. This is taking someone's blood, running it through the IM basic Duraclone panel. And as I said, this is maybe as clinical as I'm going to get today, and the using of a Duraclone. And the part of this that we were looking at is basically, okay, let's go ahead and change this to look at things that weren't necessarily what the kit was made for. In this case being the CD14 and a population of 1%. So we were glad on the SRT, we were able to translate this same Duraclone panel from our Cytoflex S right over to the SRT and really get a good look at these rare events. I also put this part up so that you can see that what you would normally see on a dot plot is this right here on the top right, the upper right quadrant, and you see all those dots, but it's the same exact that you see over here in the smaller version on the left. But the Plex platform in general on the newer software included this thing called rare events. You hit this little button and boom, it makes the dots really big. So you then, as we saw previously, you can track where they're from. This is almost the same as what we'd see if we went into our index sorting. Rare events and showing where they came from, or it's sorting into a plate, putting on a selection of what the well is, and then looking where those particular cells came from in your sort. Index sorting. I mean, anyone who's done sorting and looks at plates knows what index sorting is. For the rest of us, well, it is a very simplistic, gate a well, look to see where it came in the dot plot, and it'll put up a nice block. You can choose and give them different colors. So you can actually see each of your 96 wells in this example, where they were in this population of interest. So it's a wonderful feature that comes with most sorters now, but I think it's been very, very simplified on the SRT, as you can see here. Take that one step further. We had done with the using a Fax Aria 2U and a MoFlow Astrios, we had looked at how can we look at verification of our sorting methodology. And this was in Cytometry A, I believe, back in 2016, 17. And with my lab group, with Eric Zigan and myself, and working with Nexalom Leos Chan. And we adapted this to look now at the SRT when we first received it. So this is something I had done with my local Beckman team. And we started placing things into the wells. I put little smiley faces that we can sort into the wells. I was getting bored when I did this, if for no other reason. That's why the smiley faces are there. And here is just kind of a representation of, okay, we sorted some cells, some GFP cells onto the plate. This was a dry plate that we tried first and foremost. We put them through our Nexalom Celigo instrument. And basically then you can scour the well to see if you were able to deposit the drop. And you can see there that this is just a green bead that was sorted into, very bright green bead, obviously, that was sorted into the well. And then you go and you look in the well and you find that. The reason for that stuff around the edge is the crystallization from the droplet, because we did it in a dry well. And this is because we basically were lazy and didn't want to wait for either the beads to sink to the bottom within the fluid or have to go and try to do a lot of manipulation with some centrifugation. So we just proof of concept, put it in the dry well and image. That worked. We then again took some GFP positive cells, so some green fluorescent protein. We started putting them into the well. So here you see the initial sort. The red well that you see there on the heat map we put in there approximately 100 of the drops of the GFP positives. This was so that the machine could be focused on the actual well so that we could find the single cells a lot easier. Now, this is a twofold that really is impressive. Is one, we did. Now you can see it's telling us there's one in all these different wells. If there's more than one, it will tell us. Also, if there are none, it will show them as white in these wells with a 0. So you can actually go back and pick those particular wells and sort again into them, increasing efficiency on the wells. So as you can see in the little bar graph there, from the beginning, we were looking at an 80% efficiency. We were able to get it to 90, and then upwards of 98% efficiency, or 100%, depending on how you want to look at this resort too. We say more like 95 to 98 because some of them, there were a couple that had 2 in it, even though we hit every well. Application. So this is the part where I say a day in the life of the core, right? And how my days usually go. I didn't add meetings, calls, and things such as this. But in the general, here's what I'm doing in lab work. Here's what my colleagues are doing. This is some of the fun stuff. So we have a group, they're actually sorting right now, that do a lot of engraftment and transplantation using a basic LSK adapted methodology. And for those who are familiar with the immunological aspects of this, you have some FOXP3, so T cells, and then looking at a SLAM marker, CD150. And they are looking for CD150 lows and highs, and they sort the top 30 and the bottom 30 for their particular assays that they're running. So this is the T cells in the bone marrow that are regulatory, and looking at graft versus host type disease. And then for this one, we really just wanted to show a cool application of transplantation and graft versus host, and then also just the efficiencies of sorting those populations. So this here was the low population that we're showing, the 150 lows, and you can see here that the frequency of the lymphocyte gate was upwards of 98.5%, with the total of everything, with the slight debris that you see there being up, around 95 to 96%. So some really great results. They had been sorting on other instrumentation. We moved them to the SRT, and they have not left because it's just making their assays much easier and more productive for them. And they're actually running at pretty high speeds for what they're doing. And that was in concordance what we showed on those events per second. And you can really hone in on where you can find a very pure sample with high yield and run at a very efficient speed. All right, so this is also where we go. We have the immune response and viral resistance. So when we were saying some of my recent interests are around vaccine research, and this is all because of obviously pandemics, and whether it's we're talking about COVID-19, or we're talking about Zika viruses, or just things like allergy and inflammation. This has really started to become part of the forefront, and looking at different ways to get vaccine research done, different ways of understanding the immune response. You hear such things as those cytokine storms and all the buzzwords that came about during those 2 years of kind of pandemic lockdown. And so while it was a lockdown for most, it was really right. And so we've had some groups that have really been working on that. We've had talks with companies that are doing this research. And this is all stems from our ability to use flow cytometry to look at things like virus-like particles, VLPs, and look at extracellular vesicles or small particle detection. Here we're going to look a little bit in the larger realm. So here again is that lineage negative, SCA1 positive, c-KIT positive type platform, looking at progenitor cells. So your CMPs, MEPs, and GMPs. So all the different aspects of progenitors. You can also see where on the bottom left hand that you are looking at the progenitors, but you also can look at stem cells and separate out long terms and short terms. So you can really, If you just wanted stem cells in general, you can add another gate. You've got all your three different progenitors that are very clearly distinct. The Metaflex platform is very good at separating these out, and you can do the four-way sort and collect all those populations, and to do further downstream research is obviously the end goal. With the group, this is the Illumi group here at Beth Israel and Christina Sharkey. They look at what is basically steroid reductase. That's that SRD 5A2, and looking at how this is instrumental in kind of the prognosis of prostate cancer, and how looking at the SRD 5A2 or even down regulating it so that you can move things towards the estrogen level may either stop the growth of the prostate tumor, or be a way to kind of, if there is a marker that tells you, okay, you're starting to go into this chance of prostate cancer, or in your family history it says so. Maybe this is a way to start to, before things get into a terrible area of prostate cancer, you can kind of downregulate and start treating beforehand. So it's, this group is actually looking at those techniques, how this can be put into using like a non-invasive MRI to actually eliminate some of that expression of this steroid. Showing some of the flow. Now, I know this is not maybe the greatest, and I should have taken a ton more events in hindsight, but also show that when you're doing this type of research, you're looking at mouse models, sometimes you're not allowed a lot of cells. These are mouse getting a ton of cells to look at. And I know I'm displaying even less, as it says 20% show. And that was just to get the populations clear and really nail home that rare event kind of aspect that you're getting from this SRT that you can move right from your other instruments. And so this here is showing, okay, we can look at the luminal populations and the different luminals, whether it be L1 or L2, and the difference between those and the basal. Because a lot of times the luminum cells are going to dictate, all right, what is the prognosis and how bad is this prostate cancer? And so by using that, looking at these things, you can then go back and see if that downregulation of the steroid production within the mouse is actually being useful. So there it is again. If we're knocking it down, what are the results and how do we actually quantify that? And then by sorting out the luminum cells and the different subpopulations, can really get into kind of that systems biology thought process. This is what we were talking about. There's some Zika virus. This is some research that was done by a colleague of mine who will be presenting some more stuff. I have more of her work here also, Cecilia Cavazzoni. And even though it is an extremely Italian name, she is actually from Sao Paulo and graduated and did her PhD there at University in Sao Paulo. So when I head there, she said she is going to reach out to some of her friends. So maybe you already know her and I'm speaking to you directly, I guess. So if you do know Cecilia, seek me out when I'm there. I'd love to meet you. And if you are half as lovely as she is, it's going to be great to meet you. Another example of using that rare event type analysis button so that you can really see the events, see where they're coming from, and make better decisions on what should be sorted and what, where gating should be placed. Here is just when we say this is the antibody response to the Zika virus, and basically looking at an NS1 protein to characterize, its cross-reactivity and how your immune system responds. Search is, so they're a group that's basically looking at immune response and how this goes into many different aspects of immunology. And so obviously, again, we're talking about buzz, right? SARS-CoV-2, the spike vaccination proteins. And so we've done a lot of sorting with their group, with Cecilia and Manuel, to and Pragia to really get the B cells out. As you can see, we've done a lot of single cell B cell cloning. They're doing these BCR heavy and light chain sequencing, other kind of gene sequencing, and assessing which of these are most effective in getting memory B cells to come back and react again. And so by putting these with the supernatants of the COVID-2, you start to look at, okay, what is that immune response? And you can see here, there's some recent publications that are on that. And because I am not doing this justice whatsoever, except for the fact that the single cell sorting has been lovely and helps them pluck out the necessary germinal centers and the B cell clones they need for these studies. You can listen to Cecilia talk about that a little bit more on September 14th. You can ask your local Beckman Coulter representative for the information for that. You can probably find it on LinkedIn or any of those wonderful platforms. So, and then here is the actual kind of sort logic for some of these. Again, very limited populations that are being seen. And by being able to adjust the events, you can really hone in again. You can use that rare event portion to get in there and dig deeper. It just kind of makes your research a little bit, makes my life simpler, right? So I don't have to start guessing. Sometimes it avoids using, 10 to 20 FMOs, fluorescence -1 controls, and every pocket full of money to be spending on antibodies and every, aspect of what you should do and how you do it. So it allows to give you a little more leeway in looking and gating and getting the correct answers. Again, same group, still sorting of the clones, bringing those back and looking at how these are these B cell clones that they're able to pick out really affect the immune response when it comes to allergy and the IgE response, or your IgMs, IgGs, all the different immunoglobulin responses and just your immune response. This is also done, which we're not showing here, for transplantations, and this is in kidney transplant. Same group is doing, just different researchers taking the same thought process of B cell cloning, finding the best clone in germinal center and applying it to the different aspects, whether it be vaccination research or, as I said, allergy and inflammation and transplantation such as kidney. Another application, which is near and dear to a lot of people, and we were working with the group. Unfortunately, Monica has moved on to a different institution, but this was during the heart of the CRISPR-Cas9 kind of coming in and saying we can gene edit. And so by gene editing and picking the exact genetic edit model, they were looking at different aspects with Alzheimer's and autism, so that, neuroscience based. And even though this is a very simplistic type assay, right? This is CRISPR-Cas9 is a great thing. And it's funny that as we talk more and more immunologically about panels that can be done in 48 and 50, omit this and omit that. A lot of times in sorting, once you've done the discovery, they're really trying to narrow it down to as little as possible. And CRISPR-Cas9 kind of did that. It's 1-2 fluorescent proteins that people were looking at. And so, to be honest, this particular one that we were doing, we were having some struggles using both our normal sorting instruments that we have available. And so we moved it to the SRT, and because of the APD and being able to separate, we were getting some really good M-cherry signaling here. And even though it doesn't seem like a lot, anyone who's worked with a CRISPR-Cas9 gene editing knows that you're not getting these huge versions, but you're able to pick out those cellular populations of interest, those genetic clones, sort out those. You can then actually, culture and get more. And then basically your population becomes that 100% that you're looking for. And then you can do a lot of interesting things of looking across and understanding things about neuroscience and the astrocyte blebs here and the interaction between T lymphocytes and Tregs and what happens in things such as autism or any kind of cognitive disease, even if it goes down to things such as CTE. We're going to hit those vaccines again. Everybody loves the vaccines. They've come charging to the forefront like a bull. And so even CAR T, right, and immunotherapy, where our cancer therapies for some instances are neither as effective as we'd like, nor all inclusive. This immunotherapy and these CAR T cells have really come along, and looking at different vaccination protocols and how you can marry and merge these technologies to really get a more invasive and effective treatment for those suffering different types of cancers, and so great breakthroughs here. And so, being able to, like I said again, and I'm going to just keep hammering it home, being able to do this faster and more efficiently, really, really the science, and gets things going faster. And in healthcare, fast is instrumental and tantamount to what we want. So here, this is what one of the groups, the Avigan Lab here at Dr. Avigan is I think soon to be our cancer center chief, but it's been an intricate part of the cancer center for many years, and has built a vaccine kind of center based around this CAR T and what they're calling fusion cells. And so these dendritic cell, T cell, tumor kind of with egg-mediated here, fusion, and getting those together and giving those two individuals for hematological malignancies has really shown promise in the clinic. And so here, through a paper that they had done, is you can see from our top left, the bone marrow cells are infused. You get your mature dendritic cells. These are then labeled with a CD86. They're mixed in with this happens to be pancreatic cell line, a pancreatic tumor cell line that is M-cherry labeled. And as you get those and there's a fusogenic product, you can then sort the fusion, which happens to be a larger double positive population. And then you sort those out, and you can they gave them back to the mice with the pancreatic tumors in this instance. And you can see just some of the assays where you're doing the unstimulated and the stimulated ones, and looking at granzyme B activity or cellular killing. And so also looking at the fusogenic properties with T cells, also the CAR Ts, and how much of that is driven and what kind of survival of these T cells is there. As we know, tumors tend to really just go along and knock the immune system out. And if you're able to kind of kickstart the immune system along with attack the tumor, then you can really have a secondary kind of way of not only getting rid of the tumors, but getting rid of those kind of stealth cancer cells that may be lying still within the system that are hard to detect otherwise through whether it's flow cytometry or your simple blood test or anything of that. So you're really looking at not only getting rid of that particular malignancy, getting rid of that, what they're calling cancer, but also making sure that when you're in remission, you stay in remission. So this is what they call a cancer vaccine. And you can see, just looking at, the basic checkpoint markers, and how looking at the vaccine, you can really get some of that knockdown of the checkpoint markers and the tumor itself, the GFP labeled cells there. And then even further, when you add in these checkpoints with the vaccine, now we're building a vaccine, we're building the CAR T cells with the fusion cells and putting in the checkpoint inhibitors to really just not, like I said, again, not to just get rid of the tumor, but to make sure it does not come back in any form. And so this is, again, this synergistic vaccine approach where you're taking your fusion dendritics, adding your CAR T cells, doing T cell clonal expansions, and obliterating the tumor. And so here is those type panels where you have your fusion, your dendritic cells labeled with 11C and 86, so the double positives are what you're looking to get. And then you're basically then attaching those along with these GFP positive CAR T cells, sorting those out, and then doing functional assays, doing 10x sequencing, and getting really a protein, a gene, the antibody signature of the entire immunological response to the vaccination in this synergistic approach. Applications that we've been doing that come along. So I'm going to be honest, is when we say a day in a core, this is not just a buzz for attractiveness to get you to come listen to me talk. A lot of times a normal day, we may on the SRT, we may sit there and do each one of these type things 2 hours each. Some may be longer and take up more time. But during a normal day, we may actually see all of these projects between the hours of 9 and 6, and just keep going and getting people going with their research. So this is just, it's amazing too that how easy it is to go along. And like I said, these are just examples. And just to stop here to say, we've done bacterial analysis, we've done analysis of yeast libraries, and just all kinds of different bacteria, whether it be, rods or your normal for, you have a lot of research these days, not only just looking at bacteria in the sense of gut biome, your microbiome, looking at things for fecal transplantation, looking at, best ways to grow crops. So it's just a huge line that happen. And unfortunately, we can't even touch base on everything here. I got carried away and didn't even mention what this was. And we're looking at the humanized mouse. So engraftment and looking at model and along with human hematopoietic stem cells and do further research without having to have a bunch of human clinical patients. So as it said, this kind of humanized mouse model, it allows you to manipulate and play around with the immune system, is a desirable kind of way to also look at mutations and looking at stem cells, leukocytes, how cellular interaction. And so, the simplistic, again, kind of here is viable cells, and then let's look at those viable cells based on their basic stem cell marker, along with a protein, a fluorescent protein that we've put onto these humanized stem cells in the mouse. And we've had others that look at these things from the perspective of cardiac, arrest or infarction, myocardial infarction, and looking at in mice and representing when they release certain molecules, you can tag them human so that you know exactly where they are, that you put them in, and you can track. So again, cellular communication, cellular interaction, actual clinical application down the road. Now, what a lot of people may have come, what I've spent probably the greater part of the last 8 to 10 years trying to figure out and work with. And I've had many iterations of doing this, different instruments, different ways of doing it. And as I was having a conversation before this actual talk, we started discussing how tools are now starting to finally catch up with what we're trying to do in the research. So now, when we started, it was on a Cytoflex S. We've put some publications out. We're on multiple others. As you can see, two things, again, that came. The APD, higher sensitivity. We showed that earlier. Okay, we're showing that again. But the second, and it looks like this weird genie cloud, is blowing on these. There's APDs. It's a piezoelectric type cooling. There's also a little fan if you ever open your box, and you can see where that's underneath the pod. And you can see that as temperature decreases, okay, your dark rate or the noise that is coming from the electronics, the system, and the laser, everything starts to decrease. So your signal to noise increases. And isn't that the whole point of what you need to do, whether it's in normal cellular biology or if it's in extracellular vesicle biology? As you can increase your lasers, right? You can increase laser power, but you're going to up the noise. So how can you keep the laser power in a relatively safe state? how can you best detectors and bring that electronic noise down, increase the dynamic range, and get a better signal to noise ratio. For the separation between noise and the smallest detectable particle is essential when doing this work. And the instruments that we're using here really that somewhat more achievable than it's ever been. So I'm going to put this up. If you go onto this type website and you're doing extracellular vesicle research, okay, on the NIH NCI website that is run by Joshua Welsh in the lab of Jennifer Jones, then you are missing a lot. There is so much information. There are so many protocols and tools that really have taken and made this research attainable. And it's also made the Cytoflex an even more powerful tool in a researcher's hand. And I'm just going to show a little bit of what that actually does. What we do is we take ourselves some NIST beads. So those are the National Institutes of Standards and Technologies polystyrene beads. They give them a certification or the official thumbs up. And they give you size ranges. So these all, their size. Basically, it's a nominal size. You get a range. You get the refractive index of the bead. Everything that you need to change arbitrary units to actual nominal diameter. And this is done through FCM Pass. I also believe that part of the METVIS group with Edwin Vanderpoel and Rosetta does similar. But we tend to use the FCM Pass. And you can see there the diameters that the instrument will then calculate. And that graph is representative of those diameters. You see where it says side scatter 1 height polystyrene. Well, that is a calculation based on the violet side scatter height that are placed in a quantitative curve. So it's basically a linear regression, right? Show that linear regression here. So you can see these. This is how you look at your instrument. You can get your collection half angles. I put this one up there, even though this collection half angle does not look as nice as it should, is to show that also the power of is to show you when your instrument is not behaving. Our instrument at this point in time was not behaving necessarily like we wanted to. Called our engineer. He came, gave it the most wonderful tune-up. And next thing you know, we were looking at numbers more in the range of 57 to 58. And that is right on par with our Cytoflex S. And we knew how this data looked. You can see the scattering, the acquired data, and the regression lines, the best fit of the accuracy of the model. And then this is very important. This one's telling you your diameter based on your refractive index. And if you drop this line in as an arbitrary line, you can base basic limit of detection of my SRT. You can see that if you are biological and you are lower than 100 nanometers, I'm probably not going to see you on a scatter. But I can see 110, or as you see MESF values, I have a good chance of seeing you in a fluorescence triggering. So we started off simply by saying, well, let's mix some 100 and 150 FITSI beads together, shake them, put them on the SRT, sort them out. Now, what you're seeing here is we weren't just happy with sorting bulk. All right, we could do that. That was kind of boring, I guess. And so what we did is we decided we're going to sort these beads onto a slide into little puddles. We're going to sort 5 of the fluorescent green particles here, and we're going to sort just 1. Now, what you have to understand is here you see the dots that were seen using a 100X on a fluorescent microscope. This is a very fancy scope that our colleague, Dr. Ianitza Giron, has that helps us identify these things. The greenish spots you see in the back are because of the crystallization of the droplet, as in the microscope is not sitting right next to our SRT. You also have to put down a cover slip and whatnot. And that's the same thing that happened there. Even though this guy is arrowed out and we were looking at a focal point here in the circle, we were able to find it kind of up and out of the field of view, it's because the puddle completely dried. And so when the puddle dried, it stuck to the slide over there. But this was a great proof of concept. It worked. We proved that at least we could sort a bead. And if nothing else, we had a polystyrene small particle detector on our hands. And what's next? So we took some red blood cells. So we got ourselves a basically self-proclaimed healthy donor, and took the red blood cells. We added just an ionomycin or a complement to set the complement ring to get those out in mouse and human blood. And then basically looked and said, okay, here's where our 80 NIST beads were on in the SRT. Here's where the 100 showed up. And we just built our own little wonderful looking curve just to have while this is not completely accurate, and by no means do I say this is the way to do it, we just wanted an idea of where things would land. And to our surprise, human EVs, red blood cells, tend to look a little bit bigger than the mouse EVs. True in mouse versus human bone marrow, and also in other aspects. And what we showed here is we sorted out these human red blood cell derived EVs. And here is a wonderful electron micrograph that was done by who did this image for Dr. Guérin. And you can see here, sorted out particles, again, doing one single deposition. You can really see it and get the detail of it. And so this was great, and from our perspective, that we had something in our hands that would give us the possibility to really sort samples in an easy fashion. We've done it before. We've been on papers doing it with the MoFlow Astrios, the XDP, and others. But there's a lot that had to go into it. This is just pretty much a set it and forget it kind of methodology. And finally, what we'll see here is this was now going further. What we did is we took recombinant EVs that we purchased from Sigma. These are basically from the paper written by An Hendricks, where you're looking at viral infection of HEC293 T cells. They bleb off a bunch of recombinant EVs. And we took those, we spiked them into plasma, as you can see on the top portions here in a pre-sort. And when we looked at those spike-ins into just normal human plasma, we could find 34% GFP positive. And then we went and we sorted those. And now what we did is we looked back at our plasma with the recombinant EVs in again, and we were able to enrich that GFP positive recombinant EV population from 34 to 83%. This is really helpful in down the line analysis, in identifying cargo, and for vaccine research when you talk about virus-like particles, or looking at hydrogels or any of this kind of stuff, where you really need to get them pure and concentrated. Do the acknowledgements. So we'd like to acknowledge the individuals past and present from Beth Israel Deaconess Medical Center, Raquel and Getulio, for those of you on this talk. I didn't put those just there so I could make friends. They were actually in our lab. And so Getulio is actually coming back, and I believe Raquel is back in Rio. And then from the NIH, we mentioned Jennifer and Joshua. Our wonderful group of Beckman Coulter friends, Ken Gray, Mamadou Diallo, and Aniche Oko. Aniche, for some reason, as a rather large individual, has the hands of a saint. This guy has tuned our instruments beyond any recognition. Without him, I'm not sure we can do any EV stuff or show you wonderful separations as we did today. These are the labs that contributed their research: the Fujisaki lab, Abigan, Anderson, Illumi, Akawa, and Sage labs. Again, thanks to them for all the wonderful research they're doing and making me look like I know what I'm doing. And finally, I put on the side, join me on September 22nd, because besides detection and enrichment of microRNA and extracellular vesicles, I can't read what Raquel sent me. I get the gist. So I gave you a taste of extracellular vesicles. I gave you a taste of the possibility of detection and enrichment, but by no means is this anywhere close. This is probably a percentile. Thank you for your attention. Yes. Sorry, I have a weird sense of humor, so I like to put in, you don't have any questions, good, I can go home.
Thank you, John. Thank you very much. Thank you for the detailed presentation, and thank you for the opportunity to access sorting knowledge that you have and understand a little bit what kind of challenge you have face to face with flow users in your core facility. So thank you. And we will have more here in Brazil, right, in person. So
It's gonna be a
thank you,
lot.
including for this invitation. So we have here some couple of questions in the web chat. Let me read this. Laura, can you go down? Okay. First of all, of course, we have some questions about EVs already. So comparing the post-sorting assays between cells and vesicles, what kind of difference you see as a manager of core facility? What kind of, I mean, I'm interpreting the question.
Yeah.
What kind of difference as a manager of SRT, a sorter?
Okay, so I will kind of make it even more broad strokes of this and hopefully answer the question is, so obviously there's a lot more that goes into sorting of an extracellular vesicle as far as, I mean, we brushed over very quickly the standardization and controls that are needed to understand that you do have an extracellular vesicle population. And this was very simplistic in the sense of either pure EV populations that were purchased, or beads, things like that, that are very simple to play with. You already know what you have. A lot of the other things that will be shown later, or even some of the red blood cell EV stuff, are honed in on using orthogonal methods to really know what they are and validate that. The biggest difference too in looking at post-sort analysis especially is cells, once you get rid of debris and background, you can really get a good idea of how pure and how well you did the sort. When you start looking at things such as extracellular vesicles, that dark noise that we kind of briefly mentioned and trying to reduce that noise stays. And so you can get a perfect sort and it'll look like it. And so it's a kind of balance of how much of the noise do you cut out and how much do you keep so that people know that what you're doing is real. Because if you eliminate all the noise, questions become well, where's the noise in
it's a reference,
relation?
right?
Yeah,
it works like a reference.
It's a reference point. The other thing about noise that's very good for a reference point is it stays in the same place. It kind of has the same rate. So anything above that rate gives you the ability to understand that you now have something going through that is different than the actual instrument background. And on top of that, to add even more, I guess, confusion to it, is in theory, as we showed on the slide, if we go back quickly, on the enrichment, sometimes that noise it stays in the same place. But the other thing that happens is as that population increases, the amount of noise that is allowed to come through gets lower. So even if I just sat there and increased the number of recombinant EVs in the pre-sort, you're going to see that noise decrease because now the actual signal is greater than the noise.
Yeah, it makes sense. And you have to consider this noise if we reacquire the sorted EVs, right?
Correct. So post-sort analysis,
it will affect the percentage, right?
It is difficult. The other thing that wasn't mentioned here that I'll just give a kind of quick is you have, and I'm not going to say how we do it because you have to listen to other talks to get that. You have to concentrate out afterwards. No matter if you're using a 70 micron, a 50 micron, you're going to get more fluid than you want. And also we are not under any kind of misconception that within that droplet, because of its size compared to the EV, that there may be multiples. or contaminants. So doing whole plasma is wonderful that it can do it. The machine sees it, can work with it. But by no means does the machine know to get rid of things that are the same density and the same size per se. So that's why fluorescence becomes an invaluable tool to really increase your efficiency.
Okay, okay, thank you. Let me read the other one. John, what kind of markers do your users use in flow virometry? Can you share some examples?
Okay, so virometry, depending on what you're looking at, but a lot of these things are membrane type dyes. So those are very common in flow virometry or viral particle analysis or extracellular vesicle analysis, because they're all basic, have some sort of lipid membrane that is to them, and so using things such as dieta-NEPs, DII, DIO, DIL, cell trackers, cell masks, can give you some information. Each of them have their pluses and minuses. Most of the minuses are around the fact that they label everything that may be of a lipid nature, whether it's just your triglycerides or lipoproteins or things like that, or if you get into the fact that even viruses or bacteria can release EV-like substances. Some people find proteins of interest and then will conjugate things to that to be more particular, such as looking at like an EGFR on something, or the mouse leukemia viruses that Vera Tang had put out originally, putting something like a super folded EGFP to make them really bright and very consistent in using.
Let me read here the other question we have. Hey John, what's the biggest challenge when you compare the sorting of vesicles to cells? Okay, I already asked this. Another question. In a number day in core facility, how long it takes from turn on the equipment and get your sorted cells? In this case, cells.
Very dependent. This is all based on, so if you're just talking instrument and getting to this part where you start sorting, depending on the panels and stuff, you're talking less than an hour. And I'm not going to lie saying that this is not just me. And it may be years of experience that make it a lot easier for me to bang through these type things over and over again. But I'd still would say on a normal, say 10 color panel, you can have the instrument started and your comps and everything and hitting the sort button in approximately an hour, slightly under or over. So, plus or minus 15 minutes.
Wow.
As far as sorting goes, that's all cells you were given, how many they want back. Normally, people want what they call AMAP, or as many as possible. So, there's very few times where you're limited simply by, okay, I only need this number. Plate sorting, once it's set up and you need 96 cells, the B cell cloning, we're doing like 20 plates. 20 plates once it's set up, you're talking less than a half hour. It takes longer to take the plates in and out and treat and handle them correctly so you don't drop them on the ground than it does for the thing to just and start spitting them out. So
yeah, I can say it, I can confirm this kind of experience, but it's good to hear from a user, and it's really fast to set it up, right?
Oh yes.
Yeah. Okay, I feel that it was the last question, so thank you again, John. We are waiting for you here in São Paulo. And
yes, I will be there. I'll be there soon. If you are going to the meeting, or Raquel and you just want to hang out with me, feel free to find me while I'm there. I'll be there the 20th to the 22nd. I believe so.
We have another question here from Alessandro. Hey, John, thank you for our great presentation. Do you have practical experience sorting EVs based on size only without any staining?
Yes.
Good question.
It is a good question, and I mean the simplistic answer is yes. Um, and sometimes but again, you have to
There is a but
understand.
there. There is a
Yes,
but
because
there.
you have to understand the limit of detection. So if you say my EVs according to DLS are 60 nanometers, I can't do anything with them on a scatter. I know that I need to be somewhere in the range of 100 nanometers on a biological particle. Now that some of them may vary. This is based on thought that the refractive index is somewhere between 1.37 and 1.41. And this may be variable. I always never say no completely. I take a look first. But rule of thumb kind of is if I can't see an 80 nanometer particle polystyrene bead very clearly, I'm not going to be able to handle anything around the 100 nanometers. And so I have my instruments tuned so that I can try to accomplish that. So yes, you can do it. It's not optimal in this range. It's a lot easier to analyze them using the S. I'm not sure what happens in the flow cell because there is slight differences in the flow cells between the SRT and the S. And so you get a little better scatter resolution out of the Cytoflex S than you do the SRT. There is very clear separation between a even 100 nanometer biological, such as the red blood cell EVs, or a VLP that's about 90 compared to the SRT. A lot of those either stuck right close to the noise and you don't get a nice separation. And that's just something we've seen. So in that instance, we recommend labeling them. And most people are labeling because they have a protein of interest.
And I would say the fluorescent signal is the best we have inside of Flex platform, right? Even inside of Flex S or side of Flex BRV, right? So if you stain your EV sample, you can explore the multiparametric filters of flow cytometry. So the answer is this is answer, right? It's possible, but I would explore the staining, Alessandro.
The answer is when it comes to EVs. So when people ask about sorting EVs, the answer is there is nothing that is a kind of set. The methodology exists, the recommendations exist, but your Flex and Cytoflex SRT are going to be slightly different than mine. You may get better results. You may have a little bit of a problem. But ability to actually know that it's possible puts you in a good frame, right? Puts you where you can start and you can explore and you can really start digging into some research. And once you've figured it out, then sure, just, again, that set it, forget it, kind of put it in, run the template, and just keep getting great results. But it does take a little bit to understand how to balance the noise from the fluid, especially fluidics, right? And so we've tried a bunch of different things with the Cytoflex S, not necessarily the SRT, simply because I have a dedicated EV Cytoflex S that I call a nano flow. Whereas my SRT, like we said, and the point take home note of this talk was to say during a day, you can go from doing bone marrow cells to EVs to looking at B cell cloning in a plate, back to EV analysis with red blood cell EVs, virus-like particles. So it just becomes you've got to give a lot of love to your instrument to be able to do this much during a day. We know they're finicky, so I like to hug my SRT every morning. Let it know that I support it in its struggles of the day. So.
All right. So if we don't have any other question, I'd like to thank you again, John, and I'd like to invite everybody to come to the SBI Congress, come to our booth. We all from Beckman Coulter will be there. John will be there, and we will have his speech on September 22. So I reinforce here the invitation for you all to be there. And we have also our email, our marketing email in the web chat. So if you want to send us any other question, or if you need any other information, please contact us, and it will be a pleasure to answer you all. John, see you here in a few days.
Yes, I'm sure I'll be talking to you before then. And like you said, please join us in Sao Paulo at the SBI. And for those who want to learn a little bit more about vaccination and virus-like particles, join us on the 14th.
Okay, I'll be there.
Or if you start getting sick of me, don't join.
Okay.
Thank you,
John.
Raquel. Thank you, Flowmentor.
Have a nice evening. Bye-bye.
You also.